I want to start by just saying a couple of words about the seminar series, and then I'll hand it over to Andy. So this is the kickoff of our spring seminar series. As you all know, because I've spammed you with many emails, I've organized a special seminar this quarter centered around our Nemmers Prize winner, Kate Freeman. That is exciting for many reasons, but it also means that we have the opportunity to have some interface with the Penn State crew, including Kate herself. I see the tip of her head, and then there's a number of Penn Staters there in that owl. And so we have our two dynamic screens happening here. And so I'm hoping that through our lineup of what is actually a pretty topically broad seminar series because of the breadth of Kate's research program, we can build some of those relationships and really learn from each other and have a great quarter with everything ranging from, you know, hardcore isotope geochemistry to anthropology to all sorts of climate records of various different timescales of what I hope will be broad interest to this department. And so with that, I will turn it over to Andy as the host, and I will drop a link to what is a meeting owl in the chat, as many questions have arisen.
Excellent. Thank you, Maggie. First, I want to say congratulations to Kate. Very, very exciting. And quickly from there, say thank you, Seth, for kicking off our Nemmers Seminar series. It's very exciting, as Maggie noted. Quickly here, Seth is the Associate Director at the University of New Mexico Center for Stable Isotopes and is an Associate Professor in the Biology Department. He received his BA from Dartmouth College in 1999, and then went from there to do a PhD with Paul Koch at UC Santa Cruz, which he earned in 2005. And then from there, he held two postdoctoral positions, one with Marilyn Fogel at Carnegie and another with Carlos Martinez del Rio and Dave Williams at the University of Wyoming. Seth, I hesitate to exactly characterize how you specialize, but looking at your website, I guess, can I say isotope ecology is maybe there's so much more. Well, also let me back up further and say it's personally exciting for me to have Seth here because we have kind of inverse trajectories. About the time that I was moving from Santa Cruz to Dartmouth, he was going from Dartmouth to Santa Cruz, and we have figured out that we were on a field trip together somewhere in the late 90s, which is, you know, I guess for us is starting to feel a bit geological at this time. But for me, it was really exciting to see your name pop up on our seminar list and to go to your website and see how amazing and successful you've been since this time. And I'm very much looking forward to your seminar. And with that, I'll hand it over to you. So thank you so much.
Cool. Thanks for the introduction, Andy. So I'm presenting today from Argentina in Puerto Madryn, which is in northern Patagonia. And one of the things for living in paradise is the internet is not always the most stable. So there might be a time or two during the talk where I freeze. If that's the case, someone can pipe up and say, "Oh, will you say that again?" So anyway, I want to thanks for the introduction, and I just want to say it's a start by saying that's an incredible honor to contribute to the seminar series in celebration of Dr. Kate Freeman. She's an intellectual pioneer, as many of you know, and she's made major contributions to multiple disciplines in science, including earth sciences, anthropology, oceanography, techniques throughout her career and continues to do so, from compound-specific analysis of individual lipids to measuring the isotopic composition of elements at specific positions in a molecule. And these innovative approaches that are often applied to ancient archives have provided really unique insights into how metabolic and environmental processes have interacted over Earth history. So I really thanks for this opportunity to kick off the seminar series. It's truly a great honor, and I apologize that I couldn't be there in person. So today, I'm going to start my talk by providing a short summary of what motivates me. It's kind of like a 30,000-foot view of what our lab does and why we do it. And I'll use a recent review by Dawson et al. That argues species vulnerability to environmental change is the sum of three primary factors. It's exposure, its sensitivity, and its adaptive capacity. And while this paper focused on individual species, it's really important to remember that this framework can be easily applied to whole communities or even ecosystems. So if you plot these three factors on a single graph, a species vulnerability is essentially the inverse, species sensitivity is essentially the inverse of its adaptive capacity. And vulnerability increases positively with the species exposure and its sensitivity to environmental change. So what determines how sensitive a species is to change or how much adaptive capacity it might have is related to a combination of factors, including its life history, its genetic diversity, its phenotypic plasticity, its dispersal and colonization rates, and its resource and habitat requirements. And I've highlighted these three ones in red here because they're the focus of what we do in our laboratory. Now, another important aspect of this equation is the fact that we are currently living in an ecological bottleneck for a variety of natural and unnatural reasons. For example, what we've learned over the last century about the ecological and physiological plasticity of marine mammals, which is the reason I'm here actually in Argentina working on marine mammals, is likely a subset of their possible behavior for two main reasons. One is that most species in this group evolved and thrived during the Pleistocene, which is a period of Earth's history, as you all know, which is defined by environmental variability. And the second reason is the unnatural degree of historical human exploitation that was experienced by many marine mammals, which were hunted and sold as commodities for centuries before they were protected in the late 20th century. This persecution really reduced their population sizes to a fraction of what they were just a few hundred years ago. So going forward, as species experiences a larger array of environmental pressures, it would be good to know what species are capable of in terms of ecological and physiological plasticity, which by extension defines their adaptive capacity and may enable us to better manage them in times of rapid environmental change. So to do this, we need information on three distinct but related realms. First, I would say we need work on historical ecology that examines the ecological and physiological plasticity over evolutionary relevant timescales, okay, from centuries to millennia. So taking ecology and pushing it back in time, back at least a few thousand years, if not tens of thousands of years. An example is my dissertation work, where we reconstructed the ancient biogeography and life history of northern fur seals, and by using a combination of morphometric isotopic data, found really strong evidence that the species had extensive breeding colonies at temperate latitudes in the past, from Southern California to British Columbia, where they don't breed today. We also found that seals that bred at temperate latitudes used a very different maternal strategy, a longer lactation strategy, than their modern counterparts that mostly breed at high latitudes. And this degree of plasticity in life history had never been documented in a mammal. Another example is more recent work on North Pacific albatrosses. We found that ancient short-tailed populations spent a lot more time in the California current than their modern counterparts. And we also found that modern short-tailed albatross populations occupy a slightly higher trophic level than their ancient counterparts, probably because they interact more with industrial fisheries and they consume a lot of bycatch, which pushes their trophic level up. The second realm of work is on population and community ecology in modern ecosystems, namely what I would characterize as intra and interspecific variation in resource and habitat use. An example of this is identifying identifying the ecological and physiological causes and consequences of individual diet specialization. So we work a lot on individual diet specialization in my laboratory. And I would argue that this sort of ecological variation is a really important topic of study because it provides a real holistic measure of a species' phenotypic plasticity that integrates ecological, behavioral, and physiological factors. And it really defines what species are capable of. And by extension, it provides a really good metric for adaptive capacity and may even have evolutionary consequences. Examples in this research realm include recently published work by a postdoc in my lab, Phil Manlick, looking at the consequences of individual diet specialization in granivorous rodent community in New Mexico. And we found that generalists actually have 4 times higher annual survival than specialists. We also have examined links between individual diet specialization and phenotypic plasticity and digestive enzymes. And so this is a species of bird, the rufous-collared sparrow, that occurs throughout South America. And it shows that physiology is also a really important factor for promoting and maintaining resource inter-individual variation in resource selection. And the third work is on ecophysiology, namely what I call metabolic plasticity. That is an important component of maintaining protein homeostasis. And I really believe that physiology and ecology kind of go hand in hand. You know, physiological capabilities of organisms, how they operate, dictate their ecology and how they interact with their environment. You can't just study one or the other if you really want to understand how animals will respond to environmental change. So examples of this work include the development of methods to measure nitrogen balance in wild animals. Here we're using southern elephant seals as a model organism. And we're showing that patterns in their nitrogen isotope values of individual amino acids are driven by fasting and different biochemical processes that occur in the organism as a response to switching from exogenous resources like squid and fish to consuming your own body, consuming your muscle and fat tissue over time. And we're also developing new proxies to measure water balance in wild animals using triple oxygen isotope system, and looking at the contribution of metabolic water and how important it is for animals that live in areas where water is not all that available in terms of drinking and food water. So for today's seminar, I'll focus on projects that lie between these two realms, population community ecology and ecophysiology. You'll see here in a bit where I'm going with this. So I started out very much a population ecologist. I studied sea otter populations and northern fur seal populations through deep time. But over the past decade, I've become much more interested in community and ecosystem scale processes, specifically tracing the relative importance of different energy channels in food webs, okay? So for example, asking the question, how much kelp versus phytoplankton carbon is used to build a sea otter or sheephead in a kelp forest ecosystem? Or how does the relative availability of plant functional groups influence foraging decisions in desert small mammal communities? So how much C3 versus C4 biomass is on the landscape, and how does that influence an animal's decision-making process on what to eat for dinner? And one thing I learned while tracing energy across these ecological scales is the critical role of microbes and the role they play in transforming recalcitrant organic matter like cellulose into forms more easily assimilated by higher level consumers. And ecologists have long recognized the central role of bacteria and other microbes. That's affectionately described as ooze here in this very famous figure by Lindemann in 1942. But our ability to measure their niche has been limited until recently with the establishment of the field of microbial ecology and the widespread use of powerful genetic techniques for quantifying microbial community composition. Now, our lab has been contributing to these questions by marrying genetics with isotopes, and that enables us basically to trace the fate of microbially synthesized compounds like essential amino acids as they move up the food chain. So it turns out that food webs with multiple energy channels are more stable. Okay, fast energy channels are associated with really strong ecological interaction strengths and vice versa. So here we have fast channels depicted by phytoplankton and slow channels depicted by detritus. And this is a food web for a marine continental shelf. Now, theory and a limited amount of empirical data suggest that primary consumers specialize on distinct energy channels. So if you look at a particular ecosystem, what you see is that certain consumers focus on detrital food webs and certain consumers focus on phytoplankton-based food webs. And as you move up the food chain, okay, higher trophic level consumers actually focus on a combination of the two. And when combined with a high degree of diversity, just species diversity or richness, ecosystems with multiple slow and fast channels, as depicted here, are much more stable than those fueled by a single channel. So let's consider kelp forests as an example. This is one of my favorite ecosystems on the planet, kind of the redwoods of the ocean, which are among the most diverse marine ecosystems and are really economically important and culturally important as well. We know kelp serves as an important sort of structural role, providing habitat to many species, but how much energy actually flows from these foundation species up the food chain is an intriguing question, but one that's really difficult to answer. Of course, isotopes, specifically carbon isotopes, have been used to address this question. And the general trend is that things like phytoplankton, particular organic matter, have lower carbon isotope values than co-occurring macroalgae. Amongst the macroalgae, kelp generally has higher carbon isotope values. than red or green algae. And then seagrass, of course, has enormously high carbon isotope values. But there's often a large degree of isotopic variation and overlap in carbon isotope values among primary producers. And one reason for this is that, when you measure the isotopic composition of an autotroph primary producer, bulk tissues like leaves or fronds here, you're measuring a mixture of carbohydrates, proteins, and lipids, or a set of macromolecules. And we know that autotrophs are really good at the relative proportion of the macromolecules in their tissues changes very rapidly in response to environmental conditions and nutrient availability and light availability and temperature. And so that ability to sort of vary their macromolecular content, creates a lot of isotopic variation when you measure the bulk tissue. Another challenge is that these food webs are really complicated. Okay, so this is a kelp forest example, and it's actually a very simple kelp forest food web. But they're really rich. They contain a lot of species. They're very dynamic. And as isotopes move up food webs from the base here all the way up to the top consumers, they're sorted by physiological processes in those different types of organisms. And that sorting kind of creates noise. So you can't really, it's really difficult to sort of back calculate how much kelp carbon went into a sea otter when the carbon has to flow different levels in order to get there. And at every level, there's a lot of sort of fractionation and discrimination that can occur. So typically when ecologists measure isotopes in organisms, they measure bulk tissues, as I've mentioned. So you take a fingernail clipping from this sea otter, you take a piece of its whisker, a piece of its hair, and you'd measure largely the protein component of that particular tissue. But what my lab does is we take these protein-rich bulk tissues and we break them down into their individual amino acid or fatty acid components, okay? Because that's what proteins are, right? They're just links, links of amino acids. And we can do this, we can apply some, it's not that fancy wet chemistry. We apply some wet chemistry to this, hydrolyze the protein, and we can derivatize the amino acids, and we can measure those individual isotopic composition of those individual amino acids on the mass spectrometer. And we can measure things like non-essential amino acids, right, which are fairly simple structural forms. These are ones that animals, eukaryotes, can generally make de novo from all sorts of different substrates like carbohydrates and lipids. And then we can measure essential amino acids that eukaryotes largely have lost the ability to make because they don't have the enzymes to make them, or they don't produce the enzymes in high enough quantities to produce enough for their metabolism. So these really come from, these essentials are made de novo by plants and bacteria and fungi and protists and a few other different groups. Okay, back to basics, amino acid structure. You've all learned this at some point in your careers, high school chemistry, maybe last week in the class, or you read it in the textbook. But this is an amino acid, right? It consists essentially of what I would call a carbon skeleton, which is, the carbon backbone. And this is actually, the backbone is what an animal can or cannot make de novo. So when you say an amino acid is essential for animals, it means they cannot make this backbone, this carbon skeleton. And then an amino acid, by definition, given its name, it's got an amine group there on the end. It's nitrogen, 2 hydrogens. It's got an alpha carbon here in the middle, and it's got this carboxyl group over here. Every amino acid has that basic structure. What changes from amino acid to amino acid is what's on this R group. So in case of the simple amino acid glycine, there is no R group. Basically, take away the R and that's what you get. For something like phenylalanine, which is a very complicated structure, essential amino acid, you replace that R group with a ring, carbon ring. So there's 20 common amino acids in nature. This is just an overview. The ones that are circled in solid lines are non-essential. The dashed lines are essential. And you can see, every one of these things has an amine group, a carboxyl group over here, and then it depends on what's over here in the R group, and that's what defines the different amino acids. It's just one of my favorite depictions for showing the complexity of the different compounds. And you might ask, well, why do we focus on amino acids? Well, my answer would be that, well, we're made of them. Most animals, and that's what I am, an animal ecologist and ecophysiologist. Most 50% of the carbon in our bodies and nearly all the nitrogen in our bodies is bound in amino acids. So that's one reason why we focus on these compounds. And the other is we can lean on a really rich and detailed roadmap, so to speak, of all the biochemical interactions that occur in animals and plants or microbes to help sort of interpret the patterns in the isotopic composition that we observe. And so, just like the taxonomic tree of life, amino acids compounds have families, they have relationships, they have shared precursors, things that they're made of in these different biochemical reactions. And we lean on these things a lot. So this is just a depiction from the KEG website, one of my favorite websites that I have at the top of my bookmark bar. You can hit on this website and you can see every single biochemical interaction associated with amino acid synthesis. And we use this a lot in our lab to sort of back calculate and check out things that we're doing. So like I said, there's different challenges. Like I said, the first is this application of trophic discrimination factors. So this isotope sorting that occurs in food webs as you go up the food chain. If you really want to know, well, how much kelp, this is an Arctic food web, so there's not a lot of kelp, but let's just assume for a second, how much kelp goes into the killer whale, you have to make all these assumptions with all these different interactions. So that's a big challenge. And like I said before, the second challenge is that the isotopic composition of these primary producers at the base of the food chain, actually, they vary a lot over space and time, and they can vary very quickly. And so that's something that we have to keep in mind when we're trying to back calculate all these calculations or all these interactions. So I'm going to focus today on essential amino acids, maybe my favorite group of amino acids. I don't know, it changes weekly, but these are things that I'm going to show you data mostly through this talk for 6 different essential amino acids. And these are the ones that we can reliably measure on the mass spectrometer. And they're mostly routed from dietary protein. Like I said, animals cannot make these things de novo, so they have to get them from diet. If there's time at the end of the talk, I'm going to show you a potential other pathway that they can get them. But most of the cases, especially when you're dealing with something that has a decent amount of protein in its diet, it's getting all its essential amino acids from that diet. It's not making them de novo. And so if you can get them, you route them from your diet, that means there's minimal isotopic alteration. And this is an example of a study that's shown this. This is a controlled feeding experiment on gentoo penguins that were fed a single source. This is herring. And so what we're looking at here on the y-axis is the carbon isotope difference between the gentoo penguin and its food source, right? So for bulk tissues, you get about a 1 per mil difference, and that's what you'd expect. Here's the differences for individual non-essential amino acids over here. But what I want you to see is, or show you or highlight here, is that for the essential amino acids over here on the right, there's really not a lot of isotopic difference between the herring and the gentoo penguin, okay? So there's minimal isotopic alteration as these compounds are transferred between food and consumer. Now, the second challenge about this shifting isotopic baseline, we turn to this technique that we didn't develop, but we're certainly exploiting and further refining in our laboratory, and that is essential amino acid carbon isotope fingerprinting. And what this does is it takes advantage of the carbon isotope differences among individual amino acids in a particular sample. So here's some data. This is real data for C4 plants. These are grasses collected in New Mexico, and C3 plants. These are trees collected in New Mexico. And you can see that different amino acids, isoleucine, leucine, lysine, phenylalanine, threonine, and valine, have different carbon isotope values, right? So there's a pattern here amongst the amino acid values. And that may or may not differ between different types of organisms that can make them, okay? And now it looks like these are roughly parallel lines, so this would have the same fingerprint or the same pattern, but there's actually some subtle differences between these two patterns for C3 and C4 plants. If you compare them to patterns for fungi and bacteria, they have very, very different patterns, okay, the fungi and bacteria. So they have a very different fingerprint than do the plants shown here, okay? Now, if you stare at this, you're just like, wow, there's a lot of data there. You've got 4 sources, you've got 6 amino acids. It's hard to wrap your head around what's going on in that graph, although the patterns and whether or not those lines are parallel gives you some hints. So what we typically do is we use a multivariate analysis, like a principal component analysis or a linear discriminant analysis, to sort of take all the data for the 6 essential amino acids and be able to sort of in 2 dimensions or 3 dimensions, be able to sort of show where the major axes of variation are. Now, why does this essential amino acid fingerprinting work? Well, the short answer, and there's a much longer answer here that you could teach a whole class about, I suppose. But the short answer is that these essential amino acids are made from different precursors. There's different biosynthetic pathways between a precursor and your endpoint, the essential amino acid. So as these autotrophs and bacteria and fungi are making these things, they're using different pathways to do that. And these pathways have very different numbers of reactions. There's different things going on in those reactions. And that difference imprints on the carbon isotope value. So you get different fingerprints for different pathways. Or essentially, it's an indication of the biochemistry and that these different groups are doing the biochemistry differently. So here's a paper by Thomas Larson that came out in 2013. Thomas did a lot of the early work on this approach. So did James Scott and Marilyn Fogel at Carnegie, and Diane O'Brien, who was Thomas's postdoc advisor at Fairbanks. But if you look at this is a linear discriminant analysis. So this is a multivariate approach. This includes data for 6 essential amino acids. You've got bacteria here, you've got fungi, you've got terrestrial plants, and a variety of different marine autotrophs. And you can see that they nicely separate in this space, indicating that their essential amino acid patterns, their fingerprints, are different. You can zoom in a little bit just for the marine autotrophs, and you can see, well, seagrass are very different than the others. That makes sense. Seagrass are actually terrestrial plants that very recently invaded the oceans. But even amongst the marine producers, you've got Phaeophyceae over here, you've got microalgae, phytoplankton, you've got rhodophyte. Unfortunately, there's no green algae in this plot, but you can, if you squint your eyes, you can kind of see differences there, although there's a lot of overlap between the different groups. One thing I want to point out is that in this early compilation by Thomas, they took samples from all over the world. It was kind of like a Harmon Craig approach to doing this work, where I'll just, I'm going on a road trip, I'm going to collect samples, we're going to analyze stuff. And so, one of the questions we had was, well, at a given spot or a couple different spots, do you see these patterns? And you get better refinement in these fingerprints if you kind of control for space. So my first PhD student, now Dr. Emma Elliott Smith, did this work. She essentially wanted to create an essential amino acid library across the Pacific. She's a historical ecologist. She's interested in looking at food webs over time. She collaborates a lot with archaeologists. In fact, she's doing a postdoc now at the Smithsonian with Tori Rick, who's a coastal archaeologist. And so she collected primary producers from coastal Alaska up here, California, and northern Chile, places where kelp forests are very abundant. And she collected a variety of different primary producers: green algae, red algae, kelps, and of course phytoplankton from the different systems. Cool thing about Emma's work is because she has this kind of temporal component to ecology, she was able to collect from the Smithsonian Macrocystis porifera, which is the giant kelp here from California, and she was able to get a 120-year time series of kelp that was collected from Monterey Bay at Stanford Hopkins Marine Station. So she was able to sort of test to see how sort of variable these fingerprints were over time. Now it's only 120 years of time, but keep in mind, to an ecologist, that's a long time. That's like Earth history for an ecologist. LTERs are like 30 years long. LTERs are long-term ecological research programs, are like 30 years long. So this is a very long time scale. So in the end, she was able to do 6 essential amino acids, 125 producer samples, 2 continents, 3 oceanographic provinces, and about 120-year time frame. Now, it doesn't sound like a lot of samples, 125, but 3 years later, Emma produces this plot. Okay, so she's part of her PhD. What nicely shows is that it doesn't matter where the sites were collected or where the samples were collected, they all sort of plot on top of one another. So we have sites from Alaska, California, and Chile. The kelps all plot over here. The red algae all plots over here. Now the green algae and the phytoplankton, there's a lot of overlap. So we can't confidently discriminate between those 2 sources. But for Emma's questions having to do with kelp energy over time, this is a really nice conclusion. This is a really nice result because she can very confidently say that this kelps have a very different fingerprint than other producers in this system. And they have it over space and over time, over that 120-year time period. So she's used this data in a variety of different publications. This is just an example of one, working with colleagues in northern Chile and Antofagasta. This is the most productive marine ecosystem arguably in the world, or at least coastal ecosystem. This is part of the Peruvian current. And what you see here, actually, if you measure the bulk tissues of the different producers, that's what's down here, what you see, there's a lot of isotopic overlap between the kelps, which are here in black, and the phytoplankton, which is here in blue. Okay, so you couldn't use bulk tissues to tell you anything about energy channeling in these kelp forests. But if you do the amino acids, you see that kelps separate very nicely from the other different energy channels. Okay, and then what you can do is you can measure the fingerprints in the fish, the consumers in that food web, and the invertebrates in that food web, and you can plot those if you assume minimal isotopic alteration between the consumer and the food. You can plot those right on this same plot, and you can get an idea of what energy channels they're using most of, depending on species or individual. And one of the things from this study that was really cool was that Emma was able to show that there was specialization in energy channel usage at the individual level. So within a species, generally ecologists take a species and say, oh, you're a specialist on that, and that's what you eat. You're a specialist on that, and that's what you eat. What Emma was able to show is a lot of these species, if you take individuals within the species, they break down into different channels. So you have different individuals using different energy channels. And we think this contributes to this sort of U-shaped pattern where you get higher trophic level consumers that, contributes to this pattern that basically makes ecosystems more stable, where the higher trophic level consumers are using multiple different channels over space, and that helps with stability. So that's just one little vignette of what Emma was able to do with this library. So I want to switch gears and talk about something that's a little more closer to home in terms of where I live in the desert Southwest. This is the Rio Grande River in New Mexico. It's a beautiful landscape, actually. And we have the same sort of thing here in aquatic ecology. So now we're moving out of the oceans into freshwater ecology, and we have fast energy channels, which are the algae that's growing here in the river, things like diatoms and other algal sources, filamentous algae. And then we have slow energy channels that organisms in the river might also have access to. And those slow energy channels are terrestrial plants. So the things that live along the floodplain and the riparian ecosystems, things like willows and cottonwoods, and also a variety of non-native species like tamarisk and Russian olive, and of course, native grasses as well. Okay, but we have the same sort of serious problem. If we want to ask the question, well, how much of the fish in the river, how much of its carbon comes from in the river versus from the floodplain, right? Because these things periodically flood and this terrestrial organic matter gets inundated and then potentially transferred to the aquatic consumer. We have the same problem with this baseline, is that algae in the river has overlapping values with the dominant terrestrial plant on the floodplain, which is C3 plants. So we can't really use a bulk tissue approach to discriminate between these two sources of carbon to the food web, to the ecosystems. I also want to mention the Middle Rio Grande, like many rivers in the American Southwest, or all over the country, actually, all over the world, has experienced a lot of changes in the last 100 years. So this is in the 1920s. This is Albuquerque down here in the distance. It was a very large sort of braided stream before there was a lot of dam control and a lot of channelization that occurred. So you get this broad river that I'll show you in a second flooded a lot. So big floods, usually in the spring with snow melt up in the San Juan Mountains, and that water would inundate the floodplain, and then that would provide an opportunity for terrestrial organic matter to get entrained into the aquatic food web. So that's what the Middle Rio Grande looked like 100 years ago. This is what it looks like today. And it's basically a big channel. So we've built these levees. We've got this really nice acequia system that delivers water to different places, but we've kept the river, we've constrained the river into a very small, and its connection with this floodplain, which to be honest is this, right? My house is right in the middle of the floodplain in Albuquerque. Its connection to its floodplain has basically been severed. And so therefore a lot of opportunity for this organic carbon here, potentially important organic carbon out here on the floodplain in the form of terrestrial plants has been cut off. Now, this is just a hydrograph, again, over the last century or so, showing how the flows in the Rio Grande have decreased, okay, gone down a lot. This is probably longer term climate change that's going on. But I want to point out that the Cochiti Dam, which is upstream of Albuquerque, it's basically halfway between Santa Fe and Albuquerque, so just upstream from where we are, was put in place in the early 1970s. And what that led to is that you get a situation where, and that dam was put in place to control flooding. That was the main reason it was put in place. It wasn't for hydropower, it wasn't for recreation, it was to control flooding downstream in Albuquerque. And what you've seen happen is you've basically gone from this nice braided river where you get a lot of connection with the floodplain during seasonal pulses, flooding that occurs in the spring, to a river today that basically is just, it just stays within its channel. It's a very channelized sort of situation. You can see this a little bit. We do get floods every once in a while. We get floods here. Here's one back-to-back years, 2018 recently, where, you know, we're looking May, June, July here in the hydrograph. We get about a 500 to 600 CFS, which is a very low year. And the same time period, the next year in 2019, you go up to 4,000 to 5,000 CFS. Okay, so there is variability year to year a little bit, and you can start. And in some years, in some portions of the river, you do flood it. And so this is an example of the floodplain, the riparian ecosystem, dominated by things like willow and Russian olive and Asian elm trees. They're flooded here, and this is, again, provides an opportunity for some movement of energy back and forth. So Alexi, a PhD student in the laboratory who's finishing up, actually is interested in this. She came from an aquatic ecology background, and she wanted to ask the same question. Well, do these different sources of energy to these food webs, do they have different fingerprints? And like I mentioned, the bulk tissues, if you measure the leaves or the algae at a bulk tissue level, you get a lot of isotopic overlap. And she was able to show pretty convincingly, this is a paper that just came out this year, that, you know, different types of terrestrial plants have different fingerprints in comparison to algae. Okay, so filamentous algae are very, very different than their terrestrial counterparts that are just on the floodplain here. But Alessi is also interested in sort of microbial processes and the breakdown of that terrestrial organic matter. And if you think about it in comparison to algae, leaves are very recalcitrant. They contain a lot of cellulose. There's lignin, of course, in stems of plants and things like that. And those things aren't easily broken down by aquatic consumers. So aquatic herbivores, I'm thinking of small insects that live at the base of food chains in aquatic ecosystems. And so one thing she's interested in is what role do microbial processes play in transforming this recalcitrant terrestrial organic matter into forms of energy that are more easily used by aquatic primary consumers, like, you know, little aquatic invertebrates. So she's also added, here's these same LDAs now. She's got C3 plants, C4 plants, and there's a little bit of overlap between, she's also added in bacteria. So she was able to go out and collect bacteria from the river. She did some filtration to extract the bacteria out of the water. And then she also did some culturing. And if you look at it in 3 dimensions, there's 4 different sources here. So you actually get 3 LDA components, and you get a pretty decent separation between bacteria and algae. It doesn't look like that great in LD2, but in LD3 is actually quite good, and a little bit in LD2 as well. So anyway, she was getting a really good reclassification rate. She can fairly confidently determine whether something's an essential amino acid made by a plant versus a bacteria versus an algae using this approach. Okay, and this goes back, you know, this idea of terrestrial plants being what we call the peanut butter and cracker hypothesis, where the terrestrial plants, recalcitrant carbon is the cracker and the biofilm is the peanut butter, and this biofilm, this microbial community, essentially is transforming this terrestrial plants, this recalcitrant energy, into things like amino acids or fatty acids that are more easily used by the aquatic invertebrates. So she did a feeding experiment, basically a mesocosm study. This is the whole lab helping her out with doing all this kinds of stuff. These are just little cattle tanks. She had a couple different types of producers that she put in these tanks in the form of leaf packs. And she filled the tanks with river water and river sediment, and she just sort of let them be over a period of about 3 or 4 weeks. That sort of mimicked the spring flood that occurs usually, not always, but it used to always occur in this system. So she had cottonwood leaves, she had spiked dropseed, which is a C4 grass, and then she also used a non-native plant, which is Russian olive, that's all over the bosque. It's a very common plant and contributes a lot to the riparian biomass. Here's some data, and this is data that's sort of hot off the mass spectrometer. But here's some data looking at, again, this is for a cottonwood experiment. So these are the tanks that contain cottonwood leaf packs. And she was able to analyze both the leaf packs, but also the invertebrates that colonize those leaf packs over time. And the number in these little circles here are the numbers of weeks those leaf packs have been immersed. So the insects colonize the leaf pack, and then she's able to analyze both the leaf pack and the insects at different points in the experiment. And you see that, for cottonwood, not really all that interesting stuff going on. The leaf packs pretty much retain their cottonwood signature over time, but the invertebrates sort of start moving over time a little bit towards algae. But of all these samples, both leaf packs and invertebrates, everything classified with C3 plants. Only one individual invertebrate over here classified with algae. You can look at Russian olive. Here's the examples for Russian olive, or the data for Russian olive. And again, here's the leaf packs. They stay C3 plant over time, except one at the end of the experiment sort of migrated over towards bacteria, which is interesting. And the invertebrates very quickly actually migrate towards algae. So, these C3 plants are basically, it seems like they're forming a substrate for algal colonization. The algae gets set up on the plants, on these leaf packs, and then the invertebrates very quickly make a choice of consuming that algae rather than consuming the C3 plant. And just to show you this last bit of preliminary data for spiked dropseed, a little more interesting story going on here. Both the leaf packs and both the invertebrates are sort of migrating more towards bacteria over time, although we need a little bit more data here, and Alexi's working on that now. But this is an example of potentially, this source being routed through a microbial loop, a microbial engine, so to speak, and essentially you get the transfer of microbially synthesized essential amino acids into these aquatic invertebrates. But again, it's fairly preliminary data. I just wanted to show you kind of what we're up to and what we're doing. Another aspect of this is that, in addition to the isotopes and measuring the essential amino acids in the invertebrates and the leaf packs, Alexi is also monitoring bacterial and fungal richness over time. So she's also doing a genetic component to this mesocosm experiment. And she sees, the bacterial communities, this is the weak submerged, these bacterial communities really quickly sort of spin up and become pretty important for these different leaf packs over time. But there's a lot more work that needs to be done here to sort of figure out what these data mean. I want to quickly switch gears a little bit and talk about another sort of thing that we're interested in doing in the laboratory that has to do with mixotrophy and looking at really interesting group of animals that do both photosynthesis and they are heterotrophs as well. Okay, so that's what the definition of a mixotroph is. And we've been working a little bit on corals. And this is a really nice photograph showing, corals and cnidarian. It's an animal, right? It's basically like a sea anemone. But it has an interesting relationship, symbiotic relationship with zooxanthellae and these dinoflagellates. There's these little things in green, okay? And essentially a coral can get its nutrition from a combination of these two food sources. It can get it from the dinoflagellate via photosynthesis, or it can get it simply by filter feeding the water column and taking little bits of debris and zooplankton out of the water column and consuming it directly through its mouth here. Okay. this is work done with Mike Fox, who's now a doctor. He was a PhD student when we started this stuff. He's now a professor actually at KAUST in Saudi Arabia. And also Emma was involved in this work. We worked on corals out here on Palmyra Atoll, National Wildlife Refuge out in the middle of the South Pacific, and a specific species called the cauliflower coral, Bacillopora. And we collected it from 4 reef habitats, so not very deep, about 10 meters deep at different sort of cardinal directions of the reef. Palmyra looks like a big amoeba, basically, at 4 different sites. And we collected these things, we didn't know this at the time, but we collected them actually right before a massive bleaching event. So it's thought that this symbiotic relationship that corals have with these zooxanthellae is really important in the face of bleaching, because they have this flexibility to be able to eat like an animal or consume sugars from photosynthesis via the zooxanthellae. And what happens during bleaching events is that those zooxanthellae get ejected from the coral. And that's why corals basically turn white. It's the zooxanthellae that kind of give them that color. And they when they're bleached, when they go white, they basically eject those zooxanthellae. And the main reason they eject them is the water temperature gets too hot and the zooxanthellae leave. So it's believed that's a sort of mixotrophy, and this heterotrophic nutrition is a really important source during environmental perturbations. So if you have periods on the reef where water temperatures are really, really hot and those periods last for a long time, then the coral can lean back on heterotrophic nutrition for a little bit of time, and then wait out this environmental perturbation, this warming event, and wait for the zooxanthellae to come back, because corals don't always die after they bleach. In fact, bleaching is kind of a natural phenomenon in corals. It occurs every so often during these warming events. But the problem is those warming events are becoming more common, and they're also becoming more severe in terms of magnitude. And so, this heterotrophic nutrition sort of ability to do that is something that could be really important for determining whether or not corals can make it going forward on the planet. So you can separate the zooxanthellae really nicely from the coral animal via centrifugation, which is pretty cool. So this is just to show you an example here, what we did. This is again the LDA plot. We have phytoplankton, POM. They separate very, very differently from zooxanthellae. We didn't actually think this was going to work when we started it, but there's a really nice separation in the fingerprint between these 2 sources of nutrition for the animal. And then the animals, this sort of star shapes here, we plotted a bunch of different ones, or we analyzed a bunch of different ones from these different sites. And we find that actually, much to our surprise, corals, they eat both things. And we actually thought that essential amino acid wise, they would all come from the zooxanthellae, because if you talk to a coral ecologist, they say, well, zooxanthellae are really the main source of their nutrition. But we actually saw quite a bit of variability. And you can do a model here, and you can see about 65% comes from autotrophy. So it comes from the zooxanthellae, from an essential amino acid budget, and about 35% comes from heterotrophic nutrition. But it varies a lot amongst individuals. And some of these individuals are caught right next to each other. So different individuals in a particular place have different strategies or have the different abilities in order of using these different types of nutrition. So we get this near 0 to 100% change in their heterotrophic nutrition. And it's pretty fascinating. And again, the bulk tissue data would basically say that everything comes from the zooxanthellae. And that very little of it actually comes from the zooplankton. But we're actually seeing that for a pretty key class of compounds for corals, essential amino acids, that there's a lot more variation in where those sources are coming from, which is an interesting result. Okay, I want to finish the talk. I want to talk a little bit about green and brown energy. I think I've got about 5 minutes or so. This is work done by postdoc in the lab, actually, who just left, very sad last week. He got a position and he moved on. This is like the sad thing about being a professor is people move on. There's a lot of turnover, and I really enjoyed working with this particular individual, who I'll introduce in a second. But this is a food chain, right? So you've got primary producers here, you got primary consumers, and you got secondary consumers. This is what you see is what we call the green food web. Right? It's all based on primary production. It comes from plants or algae or something that's green because it contains chlorophyll, right? But there's also this sort of I don't know, it's a little bit more, what's the word I'm looking for? Well, it's kind of hidden, basically. But this is the brown food web. So you got to remember that about only 10% of green energy from primary producers actually goes up the food chain, because there's sort of a conversion efficiency at every single step. And only about 10% of the energy goes from plants up to primary consumers, from primary consumers to secondary consumers, and so forth. And the other 90% here goes into the brown food web, right? And it's basically decomposed by a variety of primary consumers and secondary consumers in these brown food webs, okay? And this recalcitrant green energy in this process down here in the brown food web, it gets converted by decomposers into other compounds. So microbes work at this stuff, this cellulose and this lignin, and they convert that into other compounds like amino acids and fatty acids and things that are much more usable by other consumers, even consumers that occur here in the above ground food web in this green food web. So it's possible to have amino acids that are essentially produced via decomposition here that go on to fuel the above ground food web here and to fuel the consumers that are living here that you would at first sight should be fueled only by that green energy. And interestingly, actually, there's a couple different, well, the big question here with this research is like, or one of the big questions is what regulates trophic level in food webs? So, you know, food webs have different trophic levels, different trophic steps. And the question is what are the main factors that drive those, that trophic level length? Like how many trophic levels a food chain has, which has been an open question in ecology for over 100 years. And there's been different types of hypotheses having to do with ecosystem size. This is really island stuff. Primary productivity, how much primary production goes in here drives the number of trophic levels if you have these conversion efficiencies. And then more recently, there's been some ideas, at least some empirical guesses as to how important brown energy. So if you've got brown energy that is somehow making its way back up into this above ground food web, you can actually extend the food chain length or of the above ground food web by just inserting brown energy at different points. Okay, so that's kind of this is the hypothesis that we're testing here. This is work done with Phil Manlick, who's the postdoc just left, and a colleague of mine at UNM, Joe Cook. These are sky islands around New Mexico. Okay, we're collecting 3 different species of small mammals. We have a Paramyscus maniculatus, which is largely an omnivore, and then we have 2 species of Sorex, which are insectivores, little guys that come out at night and eat insects. And we're looking at sky islands across the entire state. These are, you know, these big mountain ranges. But today I'm just going to show you some data from the Sangre de Cristo Mountains up in the Northwest, which is kind of the southern extension of the Rockies as they come into New Mexico. Here's the different potential sources. We got bacteria, we have fungi, we have plants. The ones that are open circles, we have more data right now. I just didn't have time to plot it up, but the ones that are open circles come from New Mexico, Sky Island, and the Pecos, which is in the Sangre de Cristo mountain range. And then we're comparing them with data from Larson that was collected actually in Alaska from Tulip Field Station. And we see that there's really nice agreement between plant fingerprints and fungi fingerprints. So unfortunately, we don't have any bacteria right now at this particular time, but we're working on that with cultures. And we see that you get really nice separation between these 3 potential food sources in New Mexico, as you might expect. The next thing Phil did was he looked at, he did a feeding experiment. And the main question here was to see, do organisms, essential amino acid fingerprints look like that of their diet? No one had actually tested that before. So this idea of this minimal trophic change, no one had tested it in a mammal before. I showed you data for penguins, but no one had actually done it in a mammal. He took one of our species, a colony that we have in the laboratory, and he was able to show with 100% assignment accuracy, if you feed the animal a fingerprint of soy protein, its fingerprint looks like soy protein. If you feed an animal bacterial fingerprint, its fingerprint looks like bacteria, and so on. Okay, so this is a paper that will be coming out pretty soon. We're just putting in the final revision touches on it. So here you go, you have your background again. I'm going to show you data for wild small mammals, so Paramyscus maniculatus, this omnivore, and these 2 insectivores, these 2 shrews. And there's the data, okay? And you know, you can see that there's differences between, let's say, maniculatus and monticula. There's some overlap as well, but you can take these data and again, you can put them into a mixing model and you can work out the relative proportions of green versus brown energy in these different individuals. And when you do that, this is what you get. Maniculatus, about 60% of their energy comes from plants, and another 39% comes from brown energy. For Sorex, about 50% comes from green versus brown energy, so it's about half. And as the other last species is Sorus monticola, you get about a third of their energy actually comes from plants, and most of the energy, or most of the essential amino acid budget in their bodies, comes from brown energy. Okay, so we have these different levels of green versus brown energy across these three different mammal species that occupy the same food web. And the next thing we did was we looked at those assimilated brown energy estimates, which are down here. I'll plot them on the x-axis. And then we looked at trophic level. Okay, so testing the idea that as you increase trophic level, you would get an increase in the amount of assimilated brown energy. And that's kind of what we've seen so far with our pilot data. So we get higher trophic level animals have more assimilated brown energy in them. And again, these are animals that are living above ground. They're scooting around the forest floor at night, finding insects in the above ground food web, and yet they have very high proportions of brown energy in their diets. So this really begs the question, we have to obviously do more work here, but is brown energy assimilation the ultimate driver of food chain? Okay, I think I will probably stop there. I'll finish with this slide, but basically what we're going to do, we're going to take this into the next realm is Phil, we're teaming up with some folks up in Alaska, and we're going to ask the question, with Arctic warming and the warming that's occurred over the last century or so, do you see an increase in the amount of brown energy that's available to these food webs? So this is a project that requires museum specimens. We're using, again, 3 different small mammal species as sort of model species here. We've got a high-level consumer, an insectivore, we have an herbivore and an omnivore in between. And we basically are going to be measuring the fingerprints of these animals over time to see if there's been big changes in the amount of brown versus green energy with Arctic warming. And the idea is that as the Arctic warms, more permafrost is available for decomposition, and that brown energy gets released and can be assimilated by organisms that live above ground, that are above ground consumers. And we have some pilot data here just to show you, and that is, we've got these 3 different species here. We see significant shifts between use of plants between 1990 and 2021 in the use of plants versus fungi. Bacteria don't seem to be all that important, but definitely over here in Sorex, again, we see a significant shift towards more brown energy in the more recent samples. So there's been a decent amount of warming over this 30-year time period in the Arctic, and these are all collected from the same site. And we're seeing some initial glimpse that there's been an increase in the amount of brown energy in this above ground food web over the last 30 years or so. So with that, I'm going to end, and I'll take some time for questions. And again, I just want to say it's been a wonderful, great honor to give this seminar in honor of Kate's Nemmers Prize, and I'm happy to answer any questions you have about this.